Hey guys, so today we're going to be talking about meiosis. We're going to be going over all of the different phases of meiosis, and we're going to be comparing part 1 and part 2 of meiosis. We're also going to be talking about the different processes that create genetic variation within meiosis, such as crossing over and independent assortment, and then what we end up with at the end of meiosis. So we'll start with just a definition. Meiosis is the process that we use to make gametes, which are these sex cells, sperm, and egg. It involves dividing the genetic material and the cell twice. So in meiosis, you divide the cell once. Meiosis, it happens twice. In this case, the daughter cells that are produced will have half the number of chromosomes as the parent. So we will go from a diploid cell to a haploid cell. Because of this, meiosis is sometimes called reduction division because the cells divide the number of chromosomes in half. There are two stages of meiosis, stage 1 and stage 2, and they each have 4 phases, and then cytokinesis happens at the end of each stage. Stage 1 divides the parent cell into 2 daughter cells, and then stage 2 divides each of the 2 daughter cells into 2 more cells, and that's how we end up with 4 gametes at the end. The phases of each stage are the same as each other and the same as what we would find in mitosis: prophase, metaphase, anaphase, and telophase. So we start off meiosis, which again is the way that we divide cells to make gametes. So these germ cells also take part in the cell cycle, so they will also go through interphase. They'll also have their chromosomes replicate during the S part of interphase. So you'll go from single-stranded chromatids to double-stranded chromatids with the sister chromatids attached to each other. And at that point, the germ cells will have 46 sister chromosomes. And then we'll go through stage 1, so that is prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. And then it'll go into stage 2, prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis again. So prophase 1, much like in mitosis, is going to start with the nuclear membrane breaking down and the centrioles separating and moving to opposite ends of the cell. You'll have your spindle fibers form, and the chromatin will condense into chromosomes. So all of that kind of first part of prophase 1 is the exact same as the prophase that we see in mitosis. Here's where it starts getting different. In prophase 1, all of the homologous chromosomes will pair up, so you'll have 2 of the little X's beside each other, and that's called a tetrad. Tetra meaning 4 because when there's 2 X's beside each other, there's 4 sister chromatids. That process of them actually lining up is called synapsis, and then the structure that they form when there's 2 X's together is the tetrad. And then the homologous chromosomes will actually intertwine. So you'll have like 2 X's beside each other, and the legs of the 2 separate X's will like wrap around each other. That's called crossing over. Crossing over is really important because it's one of the sources for genetic variation. So during that synapsis, the pairing up of the homologous chromosomes, non-sister chromatids, so chromatids from separate X's, will intertwine, and when they do that, they actually swap some of their alleles. Now, if you remember, the alleles are like the different options of the genes that they have on the actual chromosome. So by the end of crossing over, well, when you start crossing over, you have 2 pairs of identical sister chromatids. So you have like 2 that are the same and then another 2 that are the same. By the end of crossing over, all 4 of the chromatids will be unique. So even though you'll still have the sister chromatids attached to each other, they won't be identical anymore. So the reason for that is that one chromatid will be still all the mother's alleles, and one will be all the father's alleles. They won't have swapped any alleles. and then the two that were closest to each other when the X's were side by side will have swapped some of their alleles. So this is kind of what it ends up looking like. So this is the tetrad. So you've got the two homologous chromosomes, in this case the orange and the red, are homologous chromosomes. One is kind of the paternal version, which is the version that came from your dad. The maternal version came from your mom. So they'll pair up, and then the two in the middle, the two sister chromatids that are in the middle, will swap some of their alleles when they intertwine with each other. And so by the end, you have one sister chromatid here that's all orange, one that's mostly orange with a bit of red, one that's mostly red with a bit of orange, and one that's all red. So by the end of crossing over, all four chromatids in the homologous pair are unique. Then we go into metaphase 1, which is when the spindle fibers attach to the centromere, which is holding the double-stranded chromosome together, and it pulls each tetrad to the equator. So just like in metaphase of mitosis, you have a lining up of the chromosomes across the middle of the cell. But the difference with metaphase 1 is that instead of lining up single file like they do in normal metaphase, they're still lining up in their homologous pairs. So they're lining up 2 by 2 across the equator. This is where the second source of genetic variation is. It's called independent assortment. Essentially, it means that as they're lining up in pairs across the equator of the cell, it's going to be random which side of the equator the maternal chromosome ends up on and which side the paternal chromosome ends up on. So it won't be like all maternal on one side and all paternal on the other. It'll be like random how each pair splits up the mom and dad genes. So yeah, it means that when you divide the chromosomes, which is what's going to happen next, it'll be random where the maternal genes end up and where the paternal genes end up. Then we get into anaphase, where those spindle fibers shorten and start reeling in the chromosomes. What'll happen is that the homologous chromosomes, which were the two X's that were side by side, they are going to get pulled in opposite directions. So the sister chromatids are still attached at this point, and while attached, the two different X's are pulled to opposite sides. So this is the separation of the homologous pairs. Again, we're not separating the sister chromatids, so it's still, everything is still double-stranded at this point. This process is sometimes called segregation or disjunction because you're segregating the chromosomes to opposite ends. And by the end of this, each pole of your cell is going to have half the amount of chromosomes as what was in the middle, what it started off with. Because you had 23 pairs originally. When you separate the pairs, you now just have 23 lone chromosomes on each side. So you have 46 altogether in the middle. Now you have 23 on each side. And then we get into telophase. So you'll have around the chromosomes on each end of your cell, you'll have the nuclear membrane start to reform. The chromosomes will start to uncoil and turn back into chromatin. The spindle fibers will disappear, and the nuclear membrane will start to pinch in the middle. Then in cytokinesis, the cytoplasm will fully divide so that we end up with 2 new daughter cells. Each of those daughter cells are going to be haploid now. So we have halfway through meiosis cut the number of chromosomes in these cells in half. And our 2 daughter cells are unique, so they're not identical to each other. They're also haploid at this point. Did you say that already? I might have. Anyway. So yeah, that's just kind of what this looks like. So you still have double-stranded chromosomes, but you would have, in this case, started with 3 pairs of chromosomes in the middle, so 6 chromosomes in total in the middle. Now we have 3 on each side, and you have the pinching in of the cell membrane until it fully separates. After cytokinesis, we then go into prophase 2, where the chromatin once again condenses to form chromatin. the nuclear membrane fully dissolves again. The chromosomes are still double-stranded at this point, but you are now, you don't have homologous pairs anymore. You're down to half the number of chromosomes. The centrioles will once again move to opposite ends. Then we get into metaphase 2, which is even more similar to meiosis, mitosis. You'll notice that when we're talking about metaphase 2 and anaphase 2, they look more similar to the metaphase and anaphase of mitosis than they do to metaphase 1 and anaphase 1. So with metaphase 2, all of those double-stranded chromosomes line up single file across the equator, guided by these spindle fibers. Then during anaphase 2, you'll have segregation happening again, where you're separating things to opposite sides. This time, just like in mitosis, the sister chromatids are pulled apart and pulled to opposite ends. So we go from the double-stranded chromosome in the middle to single-stranded on either side. And then in telophase, once again, the nuclear membrane reforms around the chromosomes at either pole. The nuclear membrane will now stay that way. It's not going to dissolve again. The chromosomes will uncoil back into chromatin. The spindle fiber will go away again, and you'll have the pinching in of the cell membrane. And then with cytokinesis, that the cytoplasm and everything will divide, and you'll have 2 new cells. So at the end of the first part of meiosis, you got 2 daughter cells. Each of those 2 then go through part 2, and each of those produce 2 more daughter cells. So in the end, you end up with 4 daughter cells. Each one will be genetically unique to each other, and each one will be haploid. And by the end of this, they'll also all be single-stranded, because in part 2, we separated the sister chromatids. So this is kind of what part 2 looks like. Again, it's more similar to what mitosis looks like. So yeah, you'll have, I don't know why the title of this slide was part 1. This is part 2. This is phase 2. So you have the 2 different daughter cells that you got from the first part of meiosis. Then the chromosomes are going to line up across the middle of the cell in metaphase, and then the sister chromatids get pulled to opposite sides in anaphase, and then in telophase they all get separated. Okay, so this is what the entire process looks like. So if you look in prophase 1, we have 1, 2, 3, 4, 5, 6 chromosomes, 3 red, 3 blue. Right now they're paired up with their homologous chromosomes, so they're forming that tetrad where crossing over would happen. Then they line up 2 by 2 across the middle during metaphase 1. So they line up as homologous chromosomes. As you can see, it's random which side the blue and which side the red chromosomes go on. That's your independent assortment happening. Then the homologous pairs will get pulled apart to opposite sides. And then by the end of part 1 of meiosis, you'll have 2 separate cells. Each one will have 3 chromosomes. So we've gone from 6 down to 3 as we've separated our homologous pairs. And they're all still double-stranded. So we've gone from diploid to haploid, but we've remained double-stranded. Then we start prophase. Again, now we have 2 daughter cells. They are unique. They're not identical to each other. You compare their chromosomes, they're different colors. They will then line up during metaphase 2 across the equator. And then in anaphase 2, these sister chromatids get pulled apart. And then by the end, now you have single-stranded chromosomes. Each one of these cells still contains 3 chromosomes, so we haven't cut the number in half again. It stayed the same throughout the entire second part of meiosis, but we've gone from double-stranded to single-stranded. Okay, so this is a comparison comparing mitosis and meiosis. So with mitosis, this happens in somatic cells. The parent cell is going to be diploid, so 2N, and it's going to be double-stranded. Then you're going to have separation of the sister chromatids during the process of mitosis, which results in two identical daughter cells. Each one is still diploid, so still 2N, but they're now single-stranded. So we started diploid, we ended diploid, we just went from double-stranded to single-stranded when we separated the sister chromatids. Meiosis, on the other hand, happens in your sex cells. It's how we make our sex cells. In this case, the parent is diploid, so 2N, and double-stranded. And then during the first part of meiosis, which is what this first set of arrows represents, you have a separation of the homologous pairs. So that says homologous pairs separate. That is how we go from diploid to haploid. So we went from 2N to N, but we're still double-stranded. These are both still double-stranded. Then in the second part of meiosis, which is what this last group of arrows represents, we separate the sister chromatids. So we go from double-stranded to single-stranded. All of these last ones are single-stranded. But the number of chromosomes stays at N, so it stays haploid during that second part there. So let's just kind of zoom in on this for a second. So this is what the mitosis one looks like. Again, what we get out of it is two identical diploid cells. With meiosis, on the other hand, what we get out of it is 4 unique haploid cells. And again, this is obviously not showing the entire process here. This is just how we go from double-stranded to single-stranded. If you compare the two, you can see that both times when we separate sister chromatids, we're going from double-stranded to single-stranded, from double-stranded to single-stranded when we separate sister chromatids. The only time we go from diploid to haploid is when we separate homologous pairs, which only happens in meiosis. It doesn't happen at all in mitosis, which is why that stays diploid the whole time. Okay, let me know if you have any questions.